Hey, welcome back to the lecture on chapter 3, cell function. We just finished up with this slide on part 1, and now we're moving on to part 2, where we will focus in on the next topic, which is the cell wall. I'm bringing this one back up to remind you of the structure. So from the outside in, like the thing that's furthest out are some of these structures like flagella. Those are those long structures for movement. And then as we move in toward the cell, the next thing we encounter is the glycocalyx. And we talked about how you can have a capsule or you can have this slime layer. And then going in from there, the next structure we encounter is something called the cell wall, not to be confused with the cell membrane. The cell membrane is also known as the cytoplasmic membrane. So right now we're going to focus on the cell wall that prokaryotes have. And our human eukaryotic cells, we do not have a cell wall, though we do have a cell membrane. So focus on the cell wall. They provide structure and shape. They protect the cell from osmotic forces. That's a very fancy way of saying that this keeps the cell from swelling up too much and possibly popping, which is one of the things that humans try to do when we're trying to fight off a bacterial infection, is we're trying to get the cell to lyse. And it's the cell wall that's keeping that from happening. So we'll talk later about how some antibiotics actually function on the cell wall to allow the cell to lyse. All right, the cell wall also assists the cells in attaching to other cells, or in fact, the cell wall is somewhat protecting the bacteria against antimicrobial drugs. Because we human cells do not have cell walls, these are a great target for antibiotic drugs. Because if you have a compound that's going to affect the functioning of the cell wall or the creating of the cell wall, awesome. That's going to affect the bacterium and not going to affect us. The cell wall itself is what gives the bacteria the characteristic shapes that they have. They're made of something called peptidoglycan. Remember peptide bonds? Those are the bonds between amino acids. This is why when you connect one amino acid to another, it's called a peptide bond. When you have multiple amino acids, we call it a polypeptide. Okay, so peptido, it's telling you right in the name, there are going to be some amino acids connected to each other as part of this. And there's going to be gly. Remember what the prefix gly refers to? Sugar. So this structure is largely made of repeating sugars tied together by these little chains of amino acids. And all that matters because those are the targets of some of the antimicrobial drugs. We talk about two basic types of bacterial cell walls. It's really important in the structure. It turns out it's important. It's telling of like the evolutionary relationships, but for this class, we're not going to worry about that. We're just going to talk about this is what it looks like under a microscope. And when you stain it in the lab, it comes up with these different colors. And we'll just put them into these two categories. Why it matters is because the treatment, the antibiotics that are going to work against this kind of cell wall are different from the antibiotics that will work against this one. We refer to these two main types of bacterial cell walls as gram-positive and gram-negative. If you want a little history, gram is somebody's name. The scientist who first was trying to stain bacteria. And remember, when you look through a microscope, everything looks kind of gray. But if you start adding stains, and some structures take up the stain more than other structures do. And so the one that took up this purple stain, we called that the gram-positive one because it took that stain. And then the one that didn't take it, we call that the gram-negative one. it turns out it's kind of important in what the kind of bacteria are. And like I said, there are some of the characteristics of the infections, but this is where the name came from. Positive for taking that purple stain. Let's talk a little bit about bacterial shapes and arrangements. So these are a couple of the most common shapes and a couple of the most common arrangements. So first shapes, let's do, you've probably heard of these before, this round ball shaped bacteria. These are referred to as coccus. And then the rod shaped bacteria are referred to as bacillus. That's often part of the name of these bacteria, and the arrangement of what they, do they hold on to each other? Do they not hold on to each other? That's also pretty characteristic of species. And so we include that in the name as well. Two arrangements that I'm going to point out here are when the bacteria hold on to each other in a strip like this and like this, that look like sausages, but basically it's a strip, it's a chain. The term for that is strepto, and the term for when they're collected here, like a bunch of grapes, like disorganized bunch of grapes, that is staphylo. So this arrangement, the first one, arrangement strepto. So put the arrangement plus the shape of the cells. So this would be a streptococcus, and this would be a streptobacillus. And then finally, here's the staphylo. So this would be a staphylococcus bacterium. Now let's take a look at this cell wall that's giving us these shapes. How does it give us these shapes? What is this stuff made out of? Remember we said it's peptidoglycan. Peptido tells you peptide bonds between the amino acids, and glyco tells you sugars. Largely, so these hexagonal boxes represent the sugar units. I'm not going to go into detail on the name right now. Let's just refer to them as NAG and NAM, and we alternate NAG and NAM with each other. Once you have these long chains of the sugars, to make that a functional cell wall, they need to be held together by something. And so there are these little strips of amino acids together. Some of them are vertical. There are 4 amino acids. They're called tetrapeptide, tetra 4. It's an amino acid bridge up and down. And then side to side, there are also connecting chains of amino acids. Again, why does it matter? Because different medications affect different parts of these structures. And that's part of why in microbiology we study these structures. It's not like in and of themselves they're so fascinating. Part of it is, how can I kill this thing in a way that's not going to kill the human patient? And so that's why you need to know about these structures. All right, so we call the NAG-NAM alternating, that's the sugar backbone. And then we've got cross bridges and connecting chains. I've already mentioned the 2 main kinds, gram-positive and gram-negative. We're going to do the gram-positive ones first. This gram-positive cell wall is pretty thick, and this is the explanation, functional explanation for why did this kind of bacteria take that purple stain so well? Because this peptidoglycan layer, this very thick peptidoglycan layer, that's what takes up this purple stain. So we've got peptidoglycans. There's also some structures called teichoic acids here. And as I've mentioned, they appear purple after the gram staining procedure. And if I'm not mistaken, you will be working on the gram staining procedure in lab. Dr. Catalan is the one who kind of organizes the labs, but you'll be working with individual teaching assistants who will be teaching the labs. They'll walk you through this process of staining. As registered nurses, you're not likely to do the staining, but I think doing the procedure yourself is helpful when you get the results from the lab telling you that this patient's infection is a gram-positive bacterium or a gram-negative. It's helpful to remember what the process was for staining. There are some what are known as gram-positive bacteria. They're related to the gram-positive bacteria, but they stain a little bit differently. And the structure is up to 60% mycolic acid. That's not super important, except to know that there are some gram-positive bacteria that are referred to as acid-fast bacteria because of this acid that they're made up by, that helps them in place of this thick gram-positive bacterial cell wall. That's super important right now. This is what I want to show you. I'm going to remind you this is looking at things at 3 levels of magnification. So here's an entire bacterium, this kind of lozenge shaped things. Remember, what's that shape called? The bacillus. And we take a rectangle out of it and look at it over here. The innermost part, that's the cell membrane, the cytoplasmic membrane. And then outside of that structure is this very thick peptidoglycan layer. That's the cell wall. And this one is the gram-positive cell wall. And again, why it's gram-positive is this very thick, lots and lots of bricks. These are all the peptidoglycans, these are the NAG and NAM. It looks like little bricks. Those are the alternating NAG and NAM units. And then there's the amino acid strips that kind of hold them together. What else do I want to point out? What it looks like is outside this, there's the cell wall, and then there's a tiny little space, and then you get to the cell membrane. And just like with us, the cell membrane is made of that phospholipid bilayer. This is what in my general biology classes I would describe as the Oreo cookie layer. You've got the chocolate cookie part is the phosphate heads, both on the outside of the cell and on the inside of the cell, facing into the cell. And then the lipid tails facing each other. That's like the creamy inside part of the Oreo cookie. All right, so that's something they do have in common with us. Again, what we do not have is outside of the cell membrane, this other additional structure, the cell wall. I think I mentioned teichoic acids as structures, kind of reinforcing structures. And then there's like even the bigger ones, the lipoteichoic acids. These extend all the way through the cell wall down into the creamy inside part, the lipid part of the cell membrane. Think of these as like structural pillars. Like in your house, there's going to be some like load-bearing walls that hold up the roof. This is the roof being held up and held in position above the cell, around the cell. That was gram-positive. Now we're going to turn our attention to gram-negative bacterial cell walls. They do have peptidoglycan, like the gram-positive cell walls, but there's less of it, so not as many bricks thick. But then there's this other thing that looks kind of like a phospholipid bilayer, but it's in addition to the phospholipid bilayer on the inside. There's the gram-positive, like the gram-positive cells, there's this peptidoglycan layer. And then outside of that, there's this other, looks like phospholipid layer. It's another bilayer. It's outside of the peptidoglycan, also made of phospholipids. There are proteins. And then there's some lipopolysaccharides, LPS. So lipo means lipid, and polysaccharide, remember what that is? Poly is many and saccharide is sugars. Why this matters is that in gram-negative bacteria, there's something called lipid A. It's a structural piece of gram-negative bacteria. Coincidentally, lipid A causes all kinds of symptoms in patients. So when gram-negative bacteria have been growing in the body, as long as that lipid A portion is part of the cell, you just have an infection. But once that infection starts being treated and the gram-negative bacteria start being killed off, that lipid A is released, and that causes all manner of problem in humans. We've got the symptoms like fever, vasodilation. That means the blood vessels are going to relax, which you might think is good, brings the blood pressure down, but oh my gosh, brings it down so far that the patient's actually go into shock, which is a significant drop in blood pressure. Can cause more inflammation, which inflammation leads to more vasodilation, more leaky capillaries, and then you can get blood clotting. It's a mess. And so the treatment of the disease has to be very carefully titrated, because you want to get rid of the infection, but you don't want to kill off all of the gram-negative bacteria all at once, because that in and of itself can lead to symptoms in your patients. Okay, so under the microscope, when you look at, to detect which is your bacteria are gram-positive or gram-negative, gram-positive ones are going to look purple. And I want you to keep that word purple in your head. Then, although the gram-negative will appear pink, I'm going to ask you to replace the word pink in your brain with the word red instead, because it's like pale red. But why I'm going to ask you to do this is because it'll make it easier to remember which one's which. Purple, P, gram-positive, red, pale red, but red, negative. Like when somebody's in debt, you're in the red. So gram-negative, those are the red ones following gram staining procedure. All right, so what the structure looks like. Once again, here's that lozenge-shaped bacterium. This time, instead of a gram-positive bacterium, we're looking at a gram-negative bacterium. We take the same kind of sized rectangle out. And once again, the structure is this. On the very inside, there's the cell membrane or cytoplasmic membrane. Then there's the peptidoglycan layer of the cell wall. And if this were gram-positive, we'd be done. That's it. There's cell membrane, and then there's peptidoglycan layer, cell wall, and that's it. Gram-negative has yet another layer. So on the inside, here's that phospholipid bilayer, the Oreo cookie layer, phosphate heads, lipid tails, some embedded proteins. Then there's a little bit of space. Then there's the peptidoglycan layer, just like with gram-positive, but it's just not as thick, so it can't hold on to the stain as well, which is why it does not appear purple. But then this is what is weirder. I mean, like, this is what the gram-negatives have that the gram-positives don't have, is outside of that peptidoglycan layer, there's this additional structure. This is the outer membrane of the cell wall. And let's see, what can I tell you about that? There are these lipoproteins sticking out, and the lipopolysaccharides with a lipid A. That's the bit that is the worst news for the patient. So bad enough that the patient has an infection, right? But in treating the infection, we're going to kill off these cells. When we kill off those cells, that lipid A gets released and can start causing havoc. Fever, additional vasodilation, more inflammation. The blood pressures can tank, and the patients go into shock. Okay, I think that's what I need to cover right now with this. A couple of questions. Which of the answers below best describes the function of the bacterial cell wall? protection from all antibiotics, protection from osmotic forces, protection from temperature extremes, protection from drying out. And the correct answer to this one would be protection from osmotic forces. It keeps the cell from filling up too much with water and lysing. I did mention in passing that it may provide some protection from some antibiotics, but not all the antibiotics. And then where can lipopolysaccharide be found? That is A, in the flagella of bacterial cells, B, in the peptidoglycan layer of gram-positive bacterial cells, C, in the outer membrane of gram-positive bacterial cells, or D, in the outer membrane of gram-negative bacterial cells. And the lipopolysaccharides we're interested in are in the outer membrane of the gram-negative bacterial cells. And it's the lipid A portion that is so important. There is no such thing as the outer membrane of a gram-positive bacteria. The gram-positive cell walls just have that really thick peptidoglycan layer and no outer layer. That's unique to gram-negative cells. And then finally, there are a few bacteria that lack cell walls. They're really tiny and they're often mistaken for viruses because they're so small and they don't have a cell wall. They do, unlike viruses, they do have other features of prokaryotic cells, such as ribosomes, and that's, how we know they're actually bacteria, is they do share some structures in common with bacteria, but don't have that cell wall, but it's rare. And that is the end of this part of the lecture. See you over in part 3.